Now I have covered what could be the sampling methods, what are the various methods for cultivation of bacteria or the microbes, imaging techniques, what are the various molecular based methods, not directly the 16s but also used in the flow cytometry and used in the fluorescent in situ hybridization. Okay, that is fish. And then we have also seen genomic techniques, evolution of the genomic techniques, which has made life easy to do the metagenomic studies. Okay. Now, in the last part of the methods in microbiology, we will see that how in situ activities can be measured. Okay. So, with in situ activity, what I mean is that any analysis or any measurement you do in the nature directly. Okay, so as you can see in this particular image here, that you have a microbial mat. Okay, now in this microbial mat, what is there? There are various different kind of microbes could be, right? In the top layer you can have maybe cyanobacteria, in the bottom you can have the heterotrophic bacteria, right? And if it is present in a rock, then if you are making any study or any understanding that how much photosynthesis this cyanobacteria is making at the top, right? Or how much nitrogen is being fixed by the heterotrophic bacteria in the bottom, right? Any such a study, if you are doing, that is called as in situ activity studies. Okay, and to study, not there are not many ways, different ways. Some of them, first one is to use the microelectrodes or the biosensors. So in here, what we have is we have a sensors, micro sensors also you can say, because these are like very fine needle where the where depending on the different sort of reactions, okay, you will be able to find the you will be able to find the different sort of oxygen production, or if there is anything like nitrous oxide or pH or ammonia, nitrite, hydrogen sulfide, so and so on, right? Or carbon dioxide release. So all those things, if you can measure with the with the micro sensors, using using the electrodes or the micro electrode, right? Then that is a in situ activity, and these kind of electrodes are already present for for estimating several what do you say, physico-chemical parameters. For example, all these things what I have just mentioned here, right? And in this example you can see the partial pressure of oxygen across the day and across the depth, right? So if you do such kind of studies using the micro sensors, then that tells you the I mean in situ activity of the microbial communities. Okay, so how you can do, the first thing is by using the micro electrodes and the biosensors. Okay, I am not going in detail, but then there are several different kind of microsensors are there. For example, those microsensors which are depending on the oxidation reduction, that is mostly called as amperometric microsensors, right? Then you have this voltammetric, which depends on the generation of the voltage in the, or in the form of amperes in the sensors. Then also you have the potentiometric sensors, which measures the electrical potential at the tip, right? So all those different sort of sensors are there based on the different principles, but at the end of the day um, it will measure various gases or various for example physico-chemical conditions like pH or oxygen, right? And tells you the activity of the microbial communities. The second method to study the in situ activity is radioisotope. Now we know that in the radioisotope, what happens is that if we have a food chain and if we have fixed or if we have used the radioisotope in the primary producers, right? or otherwise in the water directly given in the medium, then that will be uptake by the primary producers. Now, if the primary producer have fixed it through the photosynthesis in the organic compound, if that organic compound is being taken by the primary consumer to secondary consumer to tertiary consumer, right? And in between, if it is releasing out in the water by the microbes, so we can analyze all the steps in that particular food chain, and then we can see that what is the structure or what is the function of the ecosystem. It means that who is feeding on whom, and how much in terms of also the quantity, right? So all those studies can be done using the radioisotope. However, radioisotopes, though they have higher energy, they are less I mean, they have very little high energy which has been used for the ecological experiments, but still they are these days less studied and has been mostly replaced with the stable isotope probes. Okay, so stable isotope probes are more heavier form of mm, uh, this isotopes. For example, carbon 13. Okay, and then you have nitrogen 15, right? It directly assimilates into the, I mean, if you give in the substrate, then it gets assimilated in the organism, and then it works in the same way how it works for the radioisotopes across the different food chain or food web. Okay, another advantage with isotope is that mostly DNA or RNA can be used as a marker. So it means, suppose if you want to study in any community that who all are doing the methanogenesis. So for that, what you do is that if you have fixed or if you have put the stable isotope in the carbon one of the methane, so what will happen? This carbon one of the methane will be used by the I mean, those organisms which are using methane, right? Methanotrophs. Now that methanotrophs will fix this carbon in the DNA as well, right? Now what will happen that the DNA where the carbon coming from the methane is fixed will have a higher density. Now, utilizing the isopycnic centrifugation, right, at a high speed in the cesium chloride gradient, you can separate that out, and then you can check that how much, I mean, which all communities have utilized, or which all species, which all individuals have utilized the methane, thereby telling you the idea about the methanotrophs in the environment, right? As the in this graph you can see that as the different isotopes have been generated, and then different development has happened, there is simultaneous development also in order to measure those isotopes. For example, in this particular case you can see that if the compound specific isotope resolutions are achieving using the different advancement, you also have at the same time utilization of flow cytometry or this nanosims, that is another form of mass spectrometry, right? Another application of the mass spectrometry, right? So all these advantages are happening in the stable isotope research. Now, stable isotope research are not only limited for understanding the microbial functioning and the food web studies, but also if you can see that where that particular stable isotope is going within the cell, so that also can be easily identified. So using the DNA based molecular biology and then in combination with the biogeochemistry, you can have the idea that what is the function or where this stable isotope is going within the cellular component, and after that in the microbial functioning and the food web studies. Not only that, the stable isotope tracers are also utilizing in the marine environment to understand the trophic interactions, right? Because because the way how the different phototrophs will fix the carbon, stable isotope carbons, right, will be varying. And then if you can differentiate that, then you can know that what fixed carbon is reaching to the host and vice versa, right? So in that way, you can find out also the stable isotope tracers of trophic interactions in the marine mutualistic studies. There is a very recent study. This is a very recent study, I guess, published in 2020. Uh, so if you are interested, you can just have a look. Another way is how you can study the microbial activity in situ is by performing mesocosm experiment. So by mesocosm, what I mean is that you do the experiment in the natural environment, and near to the natural environment, in a very big tanks. So let's say that instead of doing in 10-liter tanks in the laboratory, you are doing in 1,000-liter big drum in the marine environment itself. So those kind of experiments are more realistic in nature, and then can tell you much more information about the microbial community in the nature, rather than performing the experiment in the laboratory conditions. And again, the utilization of all these techniques, which requires the genome sequencing or protein sequencing or RNA sequencing, right, in the samples collected directly from the in situ environments. So these are some of the ways that how you study the in situ activity of the microbes. Because if you are using the metatranscriptome, you will sequence all those RNA which are expressed. So now there are more chances that if you are sequencing RNA, it means that all those are coming from the functional organisms, right? Similarly goes for the metaproteomics. So we know that after DNA there is a RNA transcription, then translation, then post-translational modifications, right? Likewise, and then only enzymatic or any physiological modifications are happening in the cell, right? So the more higher level you are studying, that is more directly linked with the activity of the organism. So it means that if you're studying DNA, that may not be directly linked with the function or the activity. If you're studying RNA, it is one step ahead to DNA. If you're studying protein, then it is one step further ahead of RNA. And then otherwise, you can also look into. There are several new methods where you can directly analyze the physiology, like metabolites and other stuff. Right. So that is how you do the in situ activity of the marine microbes.